All right. So, good afternoon, all, on this end of the weekend. And we're going to pick up right here at Neuroglia where we left off. So, throw something at her. Yeah, somebody back there is noisy. It usually implicates you. All right. Glial cells, nerve glue. So, again, we have distinctions between the four members of the central nervous system and we have the two members of the peripheral nervous system. They're all relevant in our studies today. So it's the unique idea of having a connective tissue network we see nowhere else. The nerves themselves are remarkable in the sense of the layers of protection that we'll explore. Anatomically, you already explored meningeal layers, and now we're going to see a little bit more of the microscopic construct, so to speak. So the most important and surely the most abundant of all these cells are astrocytes, star-shaped cells is how they derive their name. Highly branched, they cover literally everything, and most importantly, they cover the relationship, so they cover the capillaries and they cover the nerves themselves. And in doing so, they become an internal conduit or passageway for materials going in and out of nerves. This is one of the two intimate parts of what's called the blood-brain barrier. The blood-brain barrier means that substances in the bloodstream, unless they are small molecules which can be fat soluble or non-fat soluble but small enough, are able to pass from capillaries into the nerves. So it's very specific. Not every antibiotic can be used for neurological problems. Not every medication can be used because it doesn't. Most notably, the most famous of them all, and we'll look at it in neurotransmitters, is dopamine. Dopamine, being an amine, is polar. The dope, how we give forever, unless they've changed it dramatically, how we give people increased amounts of dopamine is we give them a precursor substance that will cross the blood-brain barrier, L-dopa, from D and L. I'm not going to go into racemic modifications and dextrorotatory and levorotatory. Isomers, remember those optical isomers? Ringing a bell? Love those. A little something that you should keep in your memory box because they're important. So by doing that, there's a combination. We have an abundance of tight junctions in the capillary structure of the capillaries that go into the brain. That's probably the primary reason why the small molecules can't get through. And as you will see shortly, illustratively, how additionally there's this layer of astrocytes that embrace both of those, and the particles literally have to pass through the astrocyte membrane, through the astrocyte, and out of the astrocyte into the nerve, and then backwards for waste products. So they are supportive, they guide development. Here's the exchange nugget, here's the development nugget as the elaborate nature of neurological tissue in both the brain and the spinal cord. They are significant with, again, what passes in and what passes out and get through the chemical environment. They will respond to various types of impulses as well as neurotransmitters. They're influential and they're certainly part of the information process. All of these are true. And generally, if it does something for the brain, it's good doing it through this. And it's a good illustration to begin to see how much it envelops and these star-shaped arms. So a particle being able to traverse the membrane of the capillary and the astrocyte can make its way kind of through the under, let's say, membrane area and back into the nerve, or leaving out in the same fashion. That's the first. Yes, sir. They, no, they are not. It's a good question. The capillaries are referred to as continuous capillaries. Fenestrated capillaries imply that they have an opening for slightly larger than smaller sized particles, whereas continuous capillaries, so it's like whenever I use this, continuous look like there's no gaps, but there's gaps. The difference here is with the abundance of tight junction, it really negates those, it's almost sealing the gaps. Fenestrated would be larger openings, so you'd begin to see a little bit, and sinusoidal would be very, very large opening. That's sort of the distinction. So they're technically a continuous capillary that you would find in a muscle or that you would find in a bone. But the reality is you don't have the liberal passage. That's there because of the abundance of the tight junctions. It has its own defense network, its own scavenger cells. They're called microglia for very tiny glial cells. Thorny processes, which is very consistent when you see what white blood cells look like. Okay, they're in touch. They go toward ones because they gobble up debris, and they can phagocytize microorganisms. And remember, the nervous system is like any other system. There are elements of it that are constantly producing waste products. So this is their representation in proximity to a nerve. So you can see all these, and these are the little appendages that we see typically with a white blood cell, because it'll have multiple appendages. It can get in touch with and engulf particles in one cell in a wide variety of areas. These are the lining cells that you will see on what are called villi. Okay, you will see these in the areas where we produce cerebrospinal fluid. They're called ependymal cells. They can be relatively flat or slightly or cuboidal to columnar. Frequently, they are ciliated, and we see them exposed into the lumen or the cavity of those wonderful ventricle areas like the third, fourth, and lateral ventricles. They cover those choroid plexes, which are really those little outpouchings of capillary material that help to process the transformation of plasma into cerebrospinal fluid, because it's not just plasma, it's modified plasma with different concentrations of electrolytes, and part of the circulatory mechanism are the ciliated cells. So you'll see them within those central cavities, lateral ventricles, third ventricles, fourth ventricle most commonly. And yeah, but they're more permeable, okay, than in other areas. So this is what they look like, and you'll see when we look at, or go back and look at chapter 12 with those choroid plexes that you'll see, or the arachnoid granulations. They'll be very similar in construction. And lastly, and as important as any, are the materials that make the myelin. And this is a big distinction. Okay. We have myelinated and non-myelinated nerves. The myelin sheaths are constructed differently, although the material is the same. The construction is different in the central nervous system, which is by way of oligodendrocytes with branches, as opposed to what you'll see in the peripheral nervous system shortly, which are Schwann cells, which are individual cells that wrap. Here, the wrapping is a little bit different. It's a larger cell that wraps multiple nerves rather than multiple single cells wrapping an individual nerve. You'll see that. So the process wraps fibers forming myelin sheaths in the thinner nerves don't require. So when we talk about white matter in the brain, it's encased, and it holds up bundles of nerves very tightly. So here you're looking at one cell, for instance, in this illustration. Here's a cell. Here it is holding, at least in this illustration, 3 different axons. And then you see, you remember what we call nodes of Ranvier, if you remember us talking about it. These are, now they call them myelin sheath gaps. They're still the same darn thing. They're just a different name. And remember, the signal effectively, even though not necessarily electrically, as you will see, as we look at the functionality, kind of jumps. We call it saltatory conjunction. So it's kind of, it goes from gap to gap, which makes the signal very fast. It insulates those areas. They don't have a lot of those, as you will see, voltage-gated sodium and potassium channels, which are clustered here. And it's the same in peripheral as well as central nerve. And we're talking about speed sometimes 100 times or more faster processing. So I like when you have a reflex and you move something fast, you touch the hot, boom. How fast do you do that? Extraordinarily quickly. That's because of this. So In the brain, we are able to package these axons closely, where in the peripheral nervous system, think going into your extremities, you have multiple nerves that are individualized, but relatively speaking, separated. They don't necessarily form in the same bundle. So now the distinction here, and for years, our discussion about satellite cells, and there's really only two. The satellite cell that surrounds cell bodies in the peripheral nervous system, I would think similar is a good word. For years, we were unsure of what it did. We think this is similar to the astrocyte. Its mission is unclear. When we talk about some regeneration in peripheral nerves, this may play a role. At least that's where the research is pointing. And then the very, very famous, they call them fancily neurolemmacytes, because the lemma is the membrane. It surrounds the membrane, the Schwann cell. All peripheral nerve fibers where myelinated sheaths are present. Okay, very much like the oligodendrocytes, and a big player. Nerves can regenerate in the peripheral nerve or the axonal segment. When as they get close to the cell body, they cannot. That's why we are able to replant limbs that have been severed if we get them in time. And the severing does not produce a lot of necrotic tissue and doesn't disturb the blood supply. I saw this at its earliest stages because when I was a resident between 1977 and 79, and nearly 50 years ago. We were just starting to see that. And we were in Philadelphia. We're in a major medical city that had, at that time, half dozen medical schools. Okay, easily, maybe more. And so there were at certain centers, the big centers, Jefferson, Hospital University of Pennsylvania, and one that we were somewhat aligned with, Hahnemann, which is now part of Drexel for you folks interested in medical school. They kind of absorbed some Hahnemann, which is down right at the corner of Broad and Vine, 14th and Vine. For you folks familiar with Philadelphia, as we like to call it. 6 years in Philadelphia. Know my way around. Not really. I need liquid. Yeah, hard stuff. Fresca. You probably put tequila in yours.
I do actually.
Why am I not shocked?
A little bit of lime juice, great day.
Just try not to do it when you're in class.
Oh, don't worry.
We were drinking in lab.
Okay, that was it. Okay, that was a gift. Okay, I would have been rude if I was. And I didn't know that you promoted ignorance. So,
just killing me. Here they are. So here you can see the sheath caps. So these are individual, and the wrapping is different. You're going to see construction-wise, this is like a jelly roll for you fans, or how you make cinnamon buns. You put the layer of dough and you put it in the middle and you wrap it up. So the nerve, and you're wrapping around. So the nucleus is close to the nerve. Okay, and if you go back and look at the illustration, it's just myelin, the way that it encases it with the oligodendrocytes in the central nervous system. So the function is the same, the construction differs. It's not a big deal, but at least it's something that's noteworthy. The nerves themselves, these, again, the part that is mitotic and reproductive, all of those glial units, that's when you hear about brain cancers, it's not the nerve. They're basically gliomas or gliosarcomas because they're connective tissue. So most brain tumors by far and away are benign. The problem is that they occupy a tremendous amount of space in an area where you don't have the space. You're in a closed cavity with a hard shell, your skull. And so when something grows in there, inevitably, it's going to push something against the wall or against something else and damage it. That is the problem. Space limitation that's there more than anything else. So the nerves themselves are amitotic. So other than maybe something that's embryological in nature. So I always get the question, well, we have brain cancer. Well, the brain cancer is the connective tissue, not the other elements that are involved. So it's kind of interesting. Uh, Optic nerve gliomas are as common as any. It's an interesting aside. You might recall me mentioning that my first wife and I had genetic testing. One of the things we tested for was Tay-Sachs disease, but the major concern for you folks who learned about this in genetics is an autosomal dominant disease, not Huntington's disease, but von Recklinghausen's disease, which she had, or has rather, called neurofibromatosis. When in one of his presentations, you're familiar with the story of a famous character in England named the Elephant Man. Okay, so if you are curious, you'll see pictures of John Merrick, and that's a presentation. What's interesting about that disease, she had type 1, that's type 4 of 5 types, and it doesn't go from generation to any offspring can change totally from the worst, the mildest to the worst presentation. There's no guarantees. So one of the things that they have are optic nerve gliomas, about 10%. She does. So one of the reasons we adopted was, did I tell you this saga? My daughter. One of the things that is a characteristic is a freckle in the eye called the Lish nodule, and that's a tell for neurofibromatosis. What do they see in my 4-year-old daughter who we adopted? No biological at all, an optic nerve. We adopted not to have nerve. They found a freckle, Lish nodule. So immediately, children's, where eventually we went here in Pittsburgh, they had to do an MRI to make sure she didn't have an optic nerve. So it's very. So I was saying, you pays your money, you takes your chances. You try to avoid potentially having a disease, and all of a sudden you're worried that adopting a child that you have absolutely no genetic biological ties could have that disease. Life is funny in that regard. And where is she today? Oh, dear, I got a text last night. We're getting on a plane 6:30. We're doing a getaway weekend in Fort Lauderdale. What is wrong with this picture? I can see you there on the beach. Yeah. So the nerves themselves, their whole job is to conduct impulses. Last your lifetime, amitotic again, very limited exceptions. And glucose. When we get to the diabetes part of the program, remember this. It's all about the glucose. They don't use any other sugar but glucose. So that's why glucose levels, particularly when they get low, are a really big problem. They have the cell, we don't call it the nucleus. The area where the nucleus is called the cell body. Remember, anatomically, the term nucleus is already used up for clusters of cell bodies. So, and then they have processes, typically two, a receptive end referred to as a dendrite, and a transmitting end referred to as an axon, which you have to know. Other names for the cell body, the soma, technically means body, and perikaryon, around the kernel or around the core. And that's where everything shakes from. Transmitters, proteins to construct neurofibrils, proteins to embedded membranes, as typically they have a certain amount of pigmentation. They're referred to as nacelles, how you pronounce that name. Chromatophilic substances in the rough endoplasmic reticulum. They have the nucleus and the nucleolus, again, the pigments. And the plasma membrane is really the critical piece with regard to it because it's both receiving and transmitting. And this is the beauty of our brain. We can get thousands of inputs, and based on those inputs, create thousands of outputs. We can assimilate all types of data. We all do it instantly. You're in a car, you're driving. You're assimilating all sorts of inputs, deciding whether I pass this person, do I get into the lane, did I put on my turning signal, did that blanky blank cut me off? Again, right? We all do that. A lot without thinking. So cell bodies, again, when we cluster nuclei, that's why we don't use the term again, and ganglia, the other term for the peripheral nervous system. So the projections, very much including their cell bodies and their processes, depending on the anatomy of it. Okay, we see them. The tracts have cell bodies more commonly in the central nervous system. Well, in the peripheral nervous system, they're just those processes, be they dendrites or axons. The term we give them is tracts with a T. Okay, in the central nervous system, and just noise. Noise. We got a lot of noise in the peripheral nervous system. Here are the processes. So this is a classic multipolar nerve. Multiple inputs. The dendrites, again, is almost this 3 branches empty of leaves. We have so many of them. So there are going to be thousands of inputs. Basically, the inputs provide information. Typically, the information is very straightforward physiologically. Here's what it says: fire or don't fire. Okay? When we get enough ones that say fire, when we reach the critical level, you and I, full threshold, it fires. They summate or add together here at a subtle rise anatomically called the axonal hillock. That's the start of the long transmitting portion, referred to as the axon. It has an initial segment, and then you have the myelination. It is unidirectional for the transmission of the action. Materials can flow in either direction. Impulses only from the cell body toward the terminus. The determinants, although it's not dramatized or illustrated particularly well, can have as many outputs as inputs, more or less. Less there, more here, less there, here. And that's by far and away the most abundant nerve in the brain, the multipolar nerve. So, hundreds of short tapering dendrites, same organelles in the cell body. Again, a lot of mitochondria because you're doing a lot of information, a lot of energy aspects that are there. And we don't use the term end plate potentials because we don't have an end plate. Okay, they're called graded potentials, and they'll either make the nerve more likely to fire, they'll be depolarizing inputs, or less likely to fire, hyperpolarizing. So, and I mean, that's how we summate. You're taking a test, and you basically look at the question, and you're thinking, what are the reasons I should answer this, and why should I pick this one, not that one? And that's what you do. All that information starts to come to the party until you figure out, aha, that's the answer. Assuming that using that kind of a logical, most of us do. So finer dendrites, very, very specialized. They have even these little spiky or bulby or knob-like microscopic appendages, called dendritic spines. So they're even more informational gathering there. And that's a really good micrograph that gives you the idea of those dendritic spines that are there. You can see the axon, you can see the hillock area. So, I mean, and again, a lot of stuff comes through. We're going to focus on, when we look at the physiology, the classic pathway, which is neurotransmitters. Nerves communicating by releasing a chemical and triggering the next nerve. It's not the only way. Sometimes they don't use neurotransmitters at all. Sometimes they don't have a connection, a synapse. There's a lot of variations that are there. So it's best to focus on these and to understand many of the others are ways we modify a response. So you and I might think of it as a filter, where you're in a group and something comes to mind, you want to say it. There's a way you're going to say it. Am I going to be loud and jump in? Am I going to wait for somebody to finish? Am I going to interrupt them? Think of the way that the group dynamics interact. That's kind of what a lot of these other nerves do. It's not, I know the answer, but how am I going to express it? Or how am I going I'm going to insert my proverbial 2 cents in, which with inflation is up to a nickel. Stop being funny. What happened to the gang here? Madison, she's on. Haha versus peculiar. My wife wouldn't say that. She made me change before I left the house. Because I was wearing the same thing I wore Wednesday.
Where's your king sweatshirt?
I was going to wear that. She said, no, those pants are horrible. You cannot go out with them. And she wasn't that friendly about it.
What she said. Like verbatim.
Oh, you're not going out in that, are you?
She's just keeping you in check. Make sure you.
It's just, just the way it was. It's okay. She had that in that kind of a tone. She has a much nicer voice than her. She still has pictures on the wall, which bothers me. She's in touch with her inner Yenzer. Believe me. She's seen the girls I was with before.
Oh, I'm sure you had to feed them off the day.
No. No.
They're probably all over you, son.
No. My wife told me I always wondered, she goes, the two things I find most attractive are wit and intelligence. And I understand that's my looks. Athletic ability? No, none.
Okay, she doesn't like that, so it's fine.
Oh, thank goodness. So, one axon that starts, there's the hillock. Length, again, 2 nerve circuits from your brain to your big toe. Depending on how tall you are, easily each one could be a meter long. So we refer to them as fibers. And again, the construction is very simple. There's multiple axons that are sheathed together. So when we say a nerve, it's not a single unit. I mean, that nerve, just like you saw at the neuromuscular junction, will have bundles of axons that will go to different motor units. Or if it's a large nerve with multiple components, like the femoral nerve or sciatic nerve, it's going to have multiple branches that go to not just one muscle, but multiple muscles, and indeed multiple parts of the muscle. So there's a lot of variation there. But that's more anatomy. They have collaterals, as you will see. And again, lots of inputs, lots of outputs. And whether you want to call them terminal branches or axonal terminals or terminal boutons, that's how you would pronounce that rather than buttons. Okay, so the axon is the transmitting or conducting, okay, along its cell membrane. So now we don't have sarcolemma, we have axonlemma. The terminal area, where how many ever there are, when you remember the neuromuscular junction images and animation, that's where those secretory vesicles that contain the neurotransmitters are located. All of that material comes from the interior of the cell down what are called neurofibrils. So they can travel again in both directions, where the impulse is unidirectional. Because you can bring materials down, you can bring waste products back up, you can reabsorb, as you will see, undergoing a process of reuptake, which is very common for many neurotransmitters. And assuming that the neurotransmitter produces the desired effect, whether it's nerve to nerve or nerve to neuromuscular junction, we may or may not have an action potential. As you can see, there's a lot of interplay that are there. And this is the other nugget about why time is an issue when there's axonal damage. And it happens all the time, but as long as you can get the ends back together. I did nothing special in surgery with regard to, I can't tell you how many patients I had. I can't feel it. It feels funny. And then a year later, it would come back. It came back because you're making a cut, and you're surely cutting tiny microscopic nerves. When you cut into the skin, or you cut into the deeper tissue, or cut a tendon or so, or lengthen, I'd cut a bone. There's all those structures are going to have damage. So it's delightful. That's why your technique in those, or anything that you do, minimizes that damage. Okay? So whether you're working on a project at home or or you're operating on someone in a very delicate environment, it's all about good technique. Three things that make a good surgeon: technique, technique, and technique. It's like real estate. Location, location, location. Yes.
It's like invisible lines, right? That surgeons try to like.
We did that. We did that. You might maybe don't recall it. There are lines of what are called relaxed skin tension. And so you know where they are. I mean, if you do mostly abdominal surgery, you know where they are in the belly. Like me, if I did mostly foot and ankle surgery, you know where they are. Now, sometimes you use them because it minimizes scar formation. And also you have an idea. You know exactly what's under there. Because if you're a resident, your attending is going, identify what's under there. Demonstrate this. Means get a hold of whatever I'm asking you. Get a hold of the extensor hallucis longus. Which tendon is it? You see three of them there. You better know it. Hopefully, so axons, internal transport. This is what I meant. Cytoskeletal elements move back and forth, and that's the idea. The good news: movement away and towards mitochondria, membranes, enzymes, neurotransmitters, cytoskeletal, backwards, degraded organelles, reuptake of molecules. Bad news: viruses and bacterial toxins. How does tetanus affect us? This way. How does botulism affect us? This way. By definition, they are neurotoxins. And there are other polio would be an example of neurotoxic bacteria. So, I beg your pardon, a neurotoxic virus. What it does is it hijacks that mechanism and is able to damage the nerve permanently. Or at least fortunately, there's a lot of nerves. So if you get to things in time, you can minimize the damage. So there's a whole lot of nuances. It's the difference, neurotoxins versus hemotoxins. So if you're talking about why certain snake bites are more dangerous than other, neurotoxins work faster. So and here we have indigenous to, what are the two venomous reptiles here in Pennsylvania? You're the one outside. What? Cottonmouth is more southern U.S. Close. Begins with a C. Copperhead. And what's the other one? Rattlesnake. Those are the two. Rattlesnake is a hemotoxin. Copperhead is a neurotoxin. The one that's more southern are the cottonmouth water moccasin. And the ones that are in the desert southwest.
We have those in PA though.
Yeah, not commonly.
Okay.
Not common. I was saying the predominant. As the coral snake, which is much more in the southwest, which is very tiny, but it's a neurotoxin. There's a great old movie from 1955 that they remade with Sean Penn, not as well, called We're No Angels, with Humphrey Bogart, Peter Ustinov, and Leo G. Carroll. It's brilliant. It's freaking brilliant. It's a Christmas movie. It's about three escapees from Devil's Island who play like three angels to help, this family that are being treated very poor. We're No Angels, 1955. It is absolutely a roar. And the star of the show is a coral snake. See, watch a little more. I started reading a biography on Moe of the Three Stooges. Slightly. That's Carly. No Three Stooges fans here? Oh, you're terrible.
All right.
You're awful. They're a law firm. Dewey, Cheatham, and Howe. Their cleaning service. They have like a dry cleaner. If you come clean with us, we'll die for you.
It's a clever. It is clever. I'll give you that.
And a lot of it's politically incorrect. There's a great one because they're all, they all play very stupid. And somebody interacts with them, and there's the three of them. They go, "You're a moron." He goes, "I'm not. I'm an idiot." And then Mo says, "He's the idiot. He's the imbecile. I'm the moron." They all take out their cards. I'm not. He's a moron. He's an imbecile.
You need to carry cards like that so people know.
Crazy. FC. That's football club.
Well, I could need something else.
Liverpool FC. So, and I forgot herpes and rabies. What was I thinking? So, and again, this is interesting. These are, again, these microRNA, and I don't know enough about them, nor would I attempt to tell you about them. But again, this underscores why in science, those that research look at these intricate things that you wouldn't think would have relevance and find ways to make them relevant. Bacteriophage, viruses being an alternative to antibiotics, this kind of thing. Which brings us to the myelin aspect, and myelin's super important. It's a whitish, it's fat and protein would be the best way to describe it. And distinctly when you see it macroscopically, it's white. When you look at a nerve in surgery, you can see the white nature. There's a lot of white structures that are there. Tendons glisten white as well. Cartilage often glistens white inside too. But they're all about transmission speed as well as protection. So the sheath, if it's sufficiently large or sufficiently long, it's myelinated. So you take the sciatic nerve, myelinated from where it originates with the multiple branches in the spinal column, but by the time it gets down, let's say to your foot, now as it gets into tiny branches that go to the intricate muscles, now it becomes non-myelinated. And even the sensory nerves that are the sensory branches of, let's say, lateral medial plantar nerve, same thing. When we would operate on them, they'd be non-myelinated, and then as we worked farther back, we could see the myelination begin. And they kind of told you where you didn't want to go, so to speak. So, and the difference is conduction speed. So the two things that really, to me, the rate determining step, which we'll get to down the road, is the synapse and the release of neurotransmitters. The other is the degree of myelination. So in the peripheral nervous system, Schwann cells, central nervous system, oligodendrocytes, here's the jelly roll thing, or whatever works for you. I like jelly rolls. Okay. So one cell is one segment. And so you'll see what it looks like. They bulge a little bit because the nucleus is there. The membranes themselves are insulators. They don't have the protein. They're mostly fatty. And so, and again, the most famous of the demyelinating disorders, other than, maybe the most common, I would think, is multiple sclerosis being damaged. We think has an autoimmune origin, but genetically, it's certainly Tay-Sachs. And that's a lysosomal storage, but what it does, effectively, it causes the myelin to deteriorate. And that's why these babies grow normally up to between age 1 and 2 and then start to deteriorate. It's very, very tragic. So here's what it looks like as it wraps around in the peripheral nervous system. You can see the location of the nucleus in this particular illustration, multiple layers around it, and always the gaps. So here it gives you an idea somewhat dramatically of a myelinated axon. That's really thick. These tiny interior these are all the various neurofibrillary tubes or neurofibrils within it. And so it's a rather distinctive and rather large. So again, now we call them sheath gaps. And this is also when we talk about collaterals, these can be inputs or outputs that modify, yes, the signal.
So the Schwann cells, they wrap around the neuron and they donate layers of their plasma membrane, and that becomes the myelin sheath?
No, their plasma membrane is the myelin sheath. Their plasma membrane wrapped around is made out of myelin.
Okay, that's
basically, if you want to call it donate, fair.
Okay.
Fair enough. But they're individual, so it's not one long sheath, or it's not multiple. So there's multiple of these in the sciatic nerve. There'll be thousands of them, and then the interspace. And we'll see this. They have very good animations that demonstrate myelinated versus non-myelinated, and you can do the conversions for the speed, which I don't do. We don't have math problems here. In the old days, I loved saying Raynaud's of Ranvier. It's like, I love the French names. Right now, when we get to immune system, we'll talk about Raynaud's disease, Raynaud's phenomena. So that's why I watched Julia, child. So not wrapped in myelin, Schwann cells, no coiling. Same deal. Central nervous system, the oligodendrocytes. You can see multiple, rather than multiple cells. Okay, it's the particular cell incorporating multiple axons. The sheath gap is there. They don't have the outer collar because the nucleus is not involved in the perinuclear cytoplasm. And that's the distinction between white matter and gray matter. When we talk about white matter, it's all the myelinated structures. Everything else, cell bodies, lots of synapses, small dendrites, variety of other structures, that's gray matter. There's the same illustration. This is the categorization or classification system that we see commonly. By far and away the most abundant, multipolar. Okay. We saw as we started to look, and we're going to pick this up again in lab because we're going to do the hearing nugget, because we did the vision nugget. Special senses frequently have bipolar. You're going to see this in the ear physiology as well as the eye. So you're taking, again, it's basically that transduction of signal from light activation by the rhodopsin and the cis transform, and now converting it to electrical signal. So you'll see it in a couple of areas that are there. Relatively rare. Nearly all sensory nerves, other than quote-unquote special sense nerves, are unipolar. Okay, and the distinction between pseudo-unipolar and unipolar has more to do with the location, okay, of the cell body. The typical unipolar nerve, when you touch something that's hot, that nerve is coming back to your spinal cord, and the cell body is very close to the dorsal area or the dorsal root area of it. So the cell body, when we come together from peripheral nerves, we call them the dorsal root ganglia. And why is that famous? Because that's the structure where the herpes viruses become latent and lead to chickenpox to shingles or herpes to recurrent. So here's the original one, multipolar. Here, the cell body is distinctly and roughly evenly placed axons and dendrites, depending on the directionality of the signal. And here, the cell body is offset from typically the axon or the longest process of it. If it's a sensory nerve, it might receive here, let's say, and bring this all the way up, and then that end will jump. Okay, that axon will transmit to the nerve within the spinal cord, or incorporate in the spinal cord and go farther. That are there, and then you can see them again here. So that's how. And now with the classification, sensory towards the central nervous system, unipolar, cell bodies again in ganglia outside, typically referred to as dorsal root, multipolar, motor rather, CNS to whatever the effector is, gland or muscle. Typically, cell bodies are in the central nervous system. When we get to the autonomic, you'll see some nuances that are there. The autonomic doesn't quite play exactly by this set of rules. It involves ganglia that are either very close to the spinal cord or ganglia that are close to or within the target organ itself. We'll get to that. Autonomic's fun. It really is a very interesting unit. And the vast majority of nerves are neither of those. The vast majority of nerves are interneurons or association neurons. Doesn't matter. 98% arbitrarily. 99%. Okay? They're the processors. You visually got a cue. What does it mean? Am I looking at your face? Am I looking at somebody who walked by the door? Am I recognizing them? Should I be responding? Is somebody trying to get my attention? All of that stuff that we're processing instantaneously is coming because of the interneuron. So it's not the absolute signal or the absolute response. It's all of the, and even when we decide what we're going to do, it's like I said, do we interrupt? Do we yell? Do I choose to raise my voice? Is it a good time to make a joke? Am I losing your attention, which is constant? That's all the stuff you do when you, sooner or later, you're going to be in front of a group in some way, shape, or form. It could be your family at the holidays, or you could be teaching a course like this. Okay, and those are all little skills. Some of us like that. I like being the center of the team. Here, I'm totally different. It's a persona. It is. I am very quiet, except in circumstances when I'm not. No. My wife throws stuff at me when I choose. She said, "You saw that terrible joke." Blank, blank.
Funny guy, Dr. Stein.
Yeah.
Funny guy.
And that concludes this presentation. Now, be wary. We're going to do the whole membrane animations, jumping from here to here type of stuff, back to the voltage gated channels and ligand gated channels and neurotransmitters forthcoming. Be ready. See you then. Have a wonderful weekend. Even have a good weekend.